Hello and welcome to CAN TV. I'm Dr. Christopher Pena. I am the scientific liaison for cancer education at the University of Chicago Medicine Comprehensive Cancer Center.
I'm Dr. Megan Mekinda. I'm the Assistant Director for Education at the U Chicago Comprehensive Cancer Center. And we have with us today a group of students from the ResearchStart program, which is a multi-institutional cancer research training program for high school students. And these amazing young scientists are working full-time all summer in a cancer research group of a faculty member at one of our four universities, the University of Chicago, the University of Illinois at Urbana-Champaign, the University of Illinois at Chicago, and Northwestern University.
Today's show is the first part of a four-part series titled Cancer Research: The Next Generation. So in this episode today, you're going to meet students who have been working at the University of Illinois at Urbana-Champaign on some really exciting innovations in cancer research and care, specifically related to bioengineering.
We know that there are many, many audience members for CAN TV who have been affected in some way by this terrible disease. And so we're really excited to be able to share with our communities some of the amazing work that's going on right here in Illinois to help prevent cancer, to help diagnose cancer, and to help treat it. To help tackle some critical issues like cancer health disparities and health equity.
So we are really thrilled to introduce you to these talented young people who, as high schoolers, are already contributing in really meaningful ways in the fight against cancer. So you are about to meet some of the newest clinicians and researchers that are going to help in this fight.
And I see your first guest is ready and eager to go, so I shall say goodbye and make some room.
So once again, we are ResearchStart. We are a research program for high school students that want to do biomedical research in the cancer field. We have several different sites where students can do this research at the University of Chicago, the University of Illinois at Chicago, the University of Illinois at Urbana-Champaign, and Northwestern University. So I am here with my first guest, Emma. Hi, Emma. How are you doing today?
I'm doing great. Thank you.
Why don't you tell us a little bit about yourself and the research that you do?
All right. So my name is Emma, as you said, and I work with monitoring angiogenesis, which is the growth of blood vessels to supply a tumor with nutrients and oxygen. So I work to monitor prostate tumor angiogenesis in mice that are lean and obese and with tomato-containing diets.
So that's really interesting. I think a lot of our viewers would kind of resonate with that because you mentioned tomato in their diets and you also mentioned lean and obese. So why are you looking at these things exactly?
Okay, so obesity, specifically an increase in adipose tissue, has been shown to promote inflammation and promote angiogenesis. And so with a tumor, that means that it's if angiogenesis is promoted, then it can grow bigger more easily because it's being supplied. So there has been a link with obesity and greater tumor and advanced prostate cancer. So we're looking at that in our obese mice, and lean we compare to see the progression of tumors. And with tomato, there has been evidence among population studies that about a cup of tomato sauce per week can reduce the risk or slow the development of prostate cancer by 30%. So it's a very realistic amount of tomato in a diet. It's not like tomato at every single meal every day. So we're using those sources of information and we're combining them together to monitor how these tumors grow.
So within these kinds of mouse models, these animals, how do you see tumors and their blood vessels?
Okay, so our method uses ultrasound imaging, and so we ultrasound mice bi-weekly, and then we switch to weekly once the tumor is detected to monitor its progression. So with us, the ultrasound imaging is normal, it's standard, we haven't really changed that at all. Of course it's up to date, but the novel thing for us is the algorithm that we run the images through after. So it uses like clutter filtering, which removes the signals we don't need from the image, and it shows the vasculature of the tumor in greater detail. So it also, with the vasculature, it also quantifies the blood power or the blood flow through that tumor, so we can see with numbers like the difference week to week.
So how does this vasculature sort of change over time?
So upon tumor detection, we see more of like a mass that's closer to a feeding vessel, we call it. So it's being fed through that already established vessel. And to continue growth, it has to have its own vasculature arise. So in our obese mice, we expect that to happen a lot faster because of that inflammation. And so we'll see those changes throughout the weeks with the ultrasound imaging.
Great. So you had mentioned before diet, obesity, and eating tomatoes, for example.
Yes.
So what's interesting is somebody can do that before they get cancer, or somebody can do that while they get cancer. Maybe the
Yes.
doctor says, hey, you need to change your diet. So with your project in particular, is this more about prevention or more about treatment?
So for the diet part of it, it is more about prevention. So like you said, changing your diet before and slowing the development of the cancer with the tomato consumption. But also with the imaging part of it, we can see the vasculature and see, oh, if the blood flow is like this, will the drugs be, will they be effective in treating the cancer? So it's both, but for the diet part, it's more about the prevention of cancer.
Wow. And, you know, everyone on this show today is coming from University of Illinois Urbana-Champaign. Can you elaborate a little bit more how the program there, specifically the engineering program, has sort of helped you on your project, which seems to be a little bit more biomedical related?
Yes. So for us, the engineering part of things has come in with the MATLAB code that we're running the images through. So that was developed by an engineering lab at UIUC, and we have basically been taught in that. The code is very user-friendly for us who aren't very familiar with it. So we run the images through that, and that's what allows us to see this quantified data, and that would not be possible without the strong engineering background at UIUC.
Awesome. Well, thanks for stopping by. It was
All
great
right.
to have you, Emma.
Thank you.
We're going to go to our next guest in just a moment, but as a reminder, we are ResearchStart. We are a program for high school students that want to partake in biomedical research over the summer. We're housed at several locations in the Chicagoland area. This includes the University of Illinois at Chicago, the University of Chicago, University of Illinois Urbana-Champaign, and Northwestern University. I am here with my next guest, Sid. Hello, Sid. How are you doing today?
I'm doing great. How
That's
are you?
awesome. Pretty good, pretty good. I heard that you work on 3D printing.
Yes.
So why don't you tell us a little bit about yourself and some of the projects that you work in with 3D printing?
So my name is Sidharth Panda. I work at the Chemical Imaging and Structures Lab under Dr. Rohit Bhargava and Mark Graika. And so I'll go a little bit in depth about 3D printing. So at its core, 3D printing is a process that can accurately recreate a tangible model of a 3D render on a computer and print it out so in a layer-by-layer basis so that you can actually interact with it.
Great. So how are you applying this sort of engineering to treat cancer?
Right. So in addition to being a chemical engineering instructors lab, chemical imaging instructors lab, we also focus on methods that help create biological structures.
Okay.
And one way that we create biological structures is by doing 3D printing with a novel type of printing method called Freeform 3D printing. And basically, we create 3D structures that can help model ducts within the human body, and help accurately model tumor microenvironments and ductal carcinomas within the human body.
So what are the benefits of using that sort of freeform 3D printing technology to create those ductal adenocarcinoma structures?
So this process is beneficial in two ways. Firstly, this method is very, very cost effective. Doctors and other researchers can quickly mass produce these structures for a very low cost, which is a much better alternative to creating a very large scale kind of version to help accurately model it within a human. And secondly is accuracy and speed. Our printer prints structures in tubes that are nearly 100 micrometers thin, which is about for reference, it's about the thickness of a strand of hair, of human hair. And this is actually just as thin as the body's actual ductal structures, and it prints those at a very, very rapid pace, around 20 seconds per structure.
Great. So we talked about this sort of really interesting technology in modeling these structures. How are you applying it in your actual experience in the lab this summer?
Right. So these 3D structures are created from a sugar called isomol, and like any sugar, if you place it within water or saline, which is kind of the which is
Solvent,
a solvent around the human body, they would very quickly dissolve. And we don't want that to dissolve because we intend to put cells, MCF10A cells, which are kind of breast cells, a human cell line, within these structures to help model, to see how these, to see the oncogenesis of these kinds of cells and the different oncogenes that would create a tumor and metastasize later. So my project is that that I'm helping to help create a spray that can help that will nebulize the structure with different materials so that the structure won't immediately deteriorate as soon as we put it in the human body. And thankfully, the versatility of our systems allows us to spray it with a whole variety of kinds of materials, from hydrophobic coatings to even proteins as well. So my project, we're looking at the most optimal kind of spray that's the most biocompatible and the most biodegradable as well when putting it in the human body. However, we don't want this, we don't want the structure to immediately dissolve at first, but we want it to dissolve at a future point in time. I think one way to kind of look at it, a nice analogy of it, is looking at a biodegradable pot that you can buy at a store. You don't want the pot to break while you're handling it,
Sure.
but as soon as you put it in the ground, you want the pot to degrade as quickly as possible so that the roots can kind of grow and the plant can make its feel around the environment, similar to how we want our MCF10A cells to grow in the human body.
That's a really interesting analogy. So sort of looking at something and trying to place it within the human body, but doing it in a careful way to not damage cells or anything like that. That's great. So now that you sort of are doing this work, what are your future plans? What's the future plans for this project, I should say, after the summer, after your data?
So in the future, we hope to grow our breast duct cells in a more complex 3D printed ductal system. So right now we kind of have a simple ductal structure, but to kind of more emulate how the ducts look within the human body, we'd like to work with more complex structures, as well as putting them in a very wide range of hydrogels, which would simulate the extracellular matrix in the human body and kind of where these structures would be. And these hydrogels would be Gelma, Pegda, Collagen 1, and much, much more. This process would ensure that our structures and our optimal compound spray that we're nebulizing our structures with are performing to the very best of their ability when placed in an environment that's similar to the human bodies.
Great. Well, thank you so much
Yeah.
for stopping by, Sid. This sounds like really compelling work, and I think one day we'll be seeing this sort of technology, hopefully, and applied in the medical field. So thank
Thank
you
you.
for stopping by. So again, we are ResearchStart. We are a program for high school students that want to partake in biomedical research over the summer, specifically within the cancer field. We are housed out of several locations in the Chicagoland area. This includes the University of Illinois at Chicago, the University of Chicago, the University of Illinois Urbana-Champaign, and Northwestern University. I am here with my last guest, Zona. Welcome, Zona.
Hi.
How are you doing today?
I'm doing well.
Great,
Yeah, thanks. How are
awesome.
you doing?
Good, good. So I heard you work with bone marrow transplants.
Yes.
And stem cells. So that's a really popular field. Why don't you share a
Yeah.
little bit about yourself and what you do with these bone marrow transplants?
Yeah, so I work in the Harley Lab at the University of Illinois in Urbana-Champaign. But generally, so what our research is tackling, or the greater problem we're dealing with is bone marrow transplants. So bone marrow transplants are used to treat patients with leukemia, a cancer of the bone marrow and the lymphatic system, as well as patients who have received ablative, chemotherapy or radiation. So research in the past few decades has capitalized on the fact that that hematopoietic stem cells, or HSCs for short, can be used for transplants to the bone marrow. So these HSCs essentially differentiate into various blood cells, lymphatic cells, and they're able to self-renew. So if you can transplant those into a patient, they can, just self-renew and differentiate into the various cells that they're missing. But there's a problem. There's still problems with if these HSCs don't engraft or essentially attach to the bones correctly, they might all start differentiating at once and not self-renewing. So this depletes the HSC pool, and all of a sudden you don't really have anything left in your body.
Well, that can't be a good thing because
Yeah,
you need
exactly.
those cells
Yeah.
to survive, I would think. So what is your specific research problem doing to kind of address this problem?
So generally what we've seen, or what researchers have seen, is that when there are higher populations of HSCs transplanted to begin with, the patients have a higher chance of, having that transplant be successful. And what we're trying to do is create a material that not only, expands and sustains a greater population of HSCs, but also mimics the natural microenvironment of the HSCs so that they're, doing all the things at the correct time, like they're self-renewing, they're differentiating, and so on.
So what particularly about the hematopoietic stem cell microenvironment are researchers really focused on?
Yeah, so bone marrow itself is actually really super complicated, but the microenvironment that these HSCs live in is called the niche, and there's a variety of different kinds of cues that are interplaying to, give signals to these HSCs about what to do. So some of these include things like biomolecular cues, such as cytokines, maybe conditions like hypoxia. There are also cues from local cell populations, such as mesenchymal stromal cells. That's another kind of stem cell in the bone marrow. Osteoblasts, the hematopoietic lineage cells, so like the different cells that the HSCs eventually differentiate into will give signals back to them. But lastly, sort of what I'm focusing on is the biophysical properties of the microenvironment, such as its stiffness, the mesh size, the degradation rate, things like stress relaxation, all also give cues to the HSCs on what to do. And the current theory sort of regarding HSCs in the bone marrow architecture is that the HSCs, which are more in a state of self-renewal, are residing closer to the bone marrow, closer to the wall of the bone, where the environment is a little bit stiffer, as well as there being other biomolecular and cellular cues, versus as those HSCs migrate towards the perivascular region.
Away,
so they're going towards the blood vessels, which will take the differentiated cells away. This environment is a little bit softer, and they're differentiating there.
Wow. So it's you sort of mentioned hardness, softness, and hydrogels. So what do hydrogels sort of do in relation to that? Like, can you adjust any properties with hydrogels, or?
Yeah, exactly. So hydrogels are really useful experimentally because there's various properties of them that you can tune. Not only that, they're really useful for clinical use later. They can, the idea is later that researchers can encapsulate HSCs in these hydrogels, transplant to the body, and then, given that you can tune the properties of the hydrogels, they're more likely to act correctly within your bone marrow when you transplant them. So what a hydrogel actually is a gelatin that is, also in water and been cross-linked to create a, sort of gelatin. So it's a gelatin, a long polymer, there's amine groups on the backbone, and we've been working with two hydrogels called GelMA and GelMal. GelMA uses methacrylyl groups added to these amines, and they can be cross-linked using UV radiation. But the problem with UV radiation isn't necessarily actually that it's dangerous, as far as research has shown. It's more that in order to penetrate all the parts of the hydrogels, which you need to cross-link, you have to keep it small and thin. So it's not scalable. And if we want to use them for microencapsulation, that's not really feasible. So GelMal has been developed exactly for that reason, so that we can have a more realistic size to encapsulate the HSCs in and expand and sustain their populations.
Right. So you had mentioned UV radiation being sort of used in the formation of cross-linkages.
Yeah.
So that UV radiation is okay for materials creation, but for people, for human beings, is it still relatively dangerous, or?
To the degree which they're using it for the HSCs, it's actually not been shown to be all that dangerous, and not all causing that, bad of an effect on the HSCs. So that's why I was saying that, that's not necessarily as big of a problem as the scalability issue. The scalability of GelMA is just not feasible for actual use.
That makes a lot of sense. So how are you sort of applying that technology in your lab this summer?
Yeah, so basically what we want to do is we want to mechanically characterize the new material, GelMal, because that allows us to then put in HSCs there and see how they react to those various mechanical properties. So the experiment I'm doing involves a compression test where you compress the samples, which are sort of like a little disc between two plans. So you push down on the sample, it measures force over time, and then you also measure the initial length of the sample, the initial height of the sample, and then you calculate two really important variables called stress and strain. This is important in materials research in general. So strain is the change of the length of the sample over the initial length, while stress is the force over a cross-sectional area of that sample. And when you plot these two variables against each other, you get a graph that sort of goes up and then stabilizes, and this is how most materials act, but to differing degrees. So the stiffness of the material is determined by the slope, and how high the slope of that initial region, the elastic region, is, and that's called, so this calculation is called Young's modulus or elastic modulus. And a lot of what I've actually been spending my time on this summer is developing a data analysis protocol for the compression data that we've been getting. So some of the data can be really messy. You don't know where necessarily zero is.
Or background.
Yeah, you have a lot of
noise,
background
all that,
noise.
yeah.
So you have to do a lot of smoothing. You have to determine where the zero is. Where does like the data actually start? So I've been developing and writing a MATLAB program to analyze that data, and by automating it, it reduces the variance between samples by eliminating the human error that you can do by just maybe going through it by hand in an Excel spreadsheet.
Oh, okay. Well, that's great.
Yeah.
It sounds like you're taking some material science and then applying a little bit of computational science.
Yeah,
to
exactly.
that as
There's
well.
a lot of different engineering fields that are at play here.
And
it's also-
biology as well.
Yeah, and biology as well. So, you're applying mechanical sciences more to biology, and I think that's really important in the future of cancer research.
Great. Well, thank you for stopping
Yeah,
by.
thanks.
Again, we are ResearchStart. We are a program for high school students that want to do biomedical research over the summer. We are housed at several locations: University of Chicago, University of Illinois Chicago, University of Illinois Urbana-Champaign, and Northwestern University. So, Zona and Sid, I'm just going to ask you a little bit about your experience here over the summer. How has University of Illinois Urbana-Champaign, particularly their engineering program, helped in your research? And then same question for you.
Yeah, so engineering in general as a field sort of seeks to design new things that can, be applied practically and, put into the commercial world and used. But there's still a lot of fundamental science that we need to understand behind that. So the University of Illinois provides a lot of opportunities to do research exactly on those things that are really important, like the fundamentals behind the things that we don't know yet. But also just the lab that I work in, the Harley lab, is extremely interdisciplinary. So we have people from a material science and engineering background to chemical engineering, chemistry, bioengineering, just biology, sort of all working together from the respective fields to bring together new ideas on the issue of cancer research, and specifically with bone marrow transplants and stem cells. And not only that, within the fields, within the research group of regenerative tissue engineering, there's multiple professors also working from even more varied fields. And just that sort of interdisciplinary culture at the university, I think, is very conducive to new and innovative ideas, which are extremely important in promoting new developments in cancer research.
So I hear interdisciplinary.
Yeah, interdisciplinary.
Sid, how do you feel about Urbana-Champaign?
I definitely agree with what Zona said. To add on, I think that it's definitely very interesting to be at the very intersection of cancer biology as well as engineering. I mean, one day in the lab I could be working on the 3D printer machine itself, and then another day, or even a couple hours from then, I could be working in a cell culture hood looking at breast cells, And I think that the interdisciplinary culture of University of Illinois Urbana-Champaign is definitely very extensive to the point that almost everything is connected with each other, and I think that's a very interesting concept to look at.
Great. Well, thank you both for coming on the show. Really excited to see what the future holds in store for both of you, and Emma as well. So that concludes our show for today. Thank you, everyone.